welcome when the next speaker is talking. We are going to move on to the second session, okay, on comparative genomics. We are going to start with Isabel Almudí from the Centro Andaluz de Biología del Desarrollo in Spain, with the presentation titled Genomic Adaptations to Aquatic and Aerial Life in Mayflies and the Origin of Insect Wings. So thank you very much. Your turn.
Thank you. Okay, so can you see my screen?
Yes.
Yeah.
Yes.
Okay, thank you. So, wait. No? Okay. So, well, good afternoon, everyone. And first of all, I want to thank Rosa and Jesus for inviting me and giving me the opportunity to talk a bit about the work we are doing with mayflies. And so in the lab, we are interested in understanding the genomic basis of morphological novelties. And for that, we are using the mayfly as a model organism. So this is a mayfly adult. And why mayflies? So first, mayflies or Ephemeroptera, together with Odonata, are the sister group to all other winged insects. So they are key to understand the origin of a morphological novelty such as is the wing of insects. Second, Cleondipteron, which is the species we work with, belongs to the Vetidae family, and this family of mayflies has a striking sexual dimorphism. So this is the female, as you saw in the previous slide, that has 2 compound eyes and 3 ocelli. But when we look at the male, what we observe is that on top of the compound eyes and the ocelli, it has another set of eyes, which are the turbanate eyes that are called like that because of this turban shape. And finally, mayflies are also quite interesting to understand adaptations to different habitats. As juveniles or nymphs, they are aquatic, they live in freshwater streams or ponds, and when they are adults, they are flying insects. So for all these reasons, we thought that it would be very interesting to explore the genome of the mayfly Cleondipteron, but for that, we need first to establish this species as a culture in the laboratory. So at the beginning, it was a bit challenging, but at the end, we managed to get our own inbred line, and we have a continuous culture running. So we have several generations that has permitted us to get the material to get a high quality reference genome. So in order to get the genome, so we sequence the genomic DNA using Illumina and nanopore strategies, but we also sequence the RNA of multiple developmental stages and tissues from nymphal or adults. And why we did that? So first, to help with the gene annotation, but also to do some analysis on different profiles of the expression pattern of these genes across the different samples. So working on that, so how these transcriptomes are expressed across the samples, and in this particular case, along the life cycle, we use MFAS subclustering software, which give us profiles of genes that are expressed along these different RNA or transcriptomes. So here, this is one example, and here you can see those genes that are highly expressed during embryogenesis, but then they are not so highly expressed during nymphal or adult stages. So this is one example, but we have many different clusters with different profiles. And here, then we wanted to focus in this transition between the aquatic and the adult stage. So we have here one cluster, the cluster 18, which has genes with high expression only in the nymphal stages, versus this other cluster, cluster 30, which has genes that have a high expression during the adulthood. And when we look the type of genes and using gene ontology term enrichment analysis, we see which type of genes are those in these two clusters. And for example, for the aquatic phases, what we find are genes that are involved in chemical stimuli perception or odorant binding, whereas when we look at the genes that are in this cluster and the terms that are enriched are always or mostly related to visual perception. So something that we like to say in the what we like to say in the lab is that the nymphs smell, but the adults see the external environment. So we wanted to go a bit more into the details of the chemoperception, and something that is important to take into account before is that the transition from water to land really requires some physiological, anatomical, and molecular adaptations in the chemosensory systems. So in insects, there are several chemosensory gene families, but there are two that are specific of insects. So the odorant binding proteins, which appear for the first time in Hexapoda, and the odorant receptors that appear in Insecta. So these two gene families have been always suggested to appear as an adaptation of the terrestrial life of insects, in contrast to crustaceans.
Sorry, Isabel, I needed to mute everyone, so feel free to unmute yourself and continue the talk. Isabel?
Now?
Yes, thank you.
Okay. Sorry. Okay, so I was saying that that these genes, these gene families have been proposed as an adaptation to terrestrialization of insects. So we wanted to explore what happened or what happens in Cleondipterum, and for that we went back to our MFAS clusters, and we detected that most of the chemosensory genes were included in 4 clusters. And when we looked which type of clusters were there, we observed that most of them were clusters associated to the aquatic stages of the life cycle, except for this one, the cluster 10, that belongs to genes, so genes that just have a peak of expression before hatching into the water. So moreover, we also look at particular tissues, and in particular the odorant binding proteins, and we saw that most of them were highly expressed in nymphal heads, so aquatic nymphal heads again, but there were a second tissue in which some of them were highly expressed, which were the gills. So the gills of the mayfly have been always described as respiratory organs. So here what we are seeing is that these gill plates are not only functioning as a respiratory organ, but they also function as a sensory organ. As when we check the expression of some of these odorant binding proteins in the gill plate, we observe expression throughout all over the gill, but this expression colocalized with the neural marker HRP, really indicating that so these gill plates also have a role in a sensory function. So this is what we explored about the chemoperception. So what happens to the visual perception in the adults? So in terms of vision, in insects, there are 3 main light sensing opsins: the long wave sensitive opsin, the blue sensitive opsin, and the ultraviolet sensitive opsin, that have undergone different expansions or reduction independently in the different lineages of insects. So what happens to Cleondipterum? So we also observe expansions of the long wave sensitive opsin, something that is quite common in other lineages, but what was particularly interesting in the case of Cleon is the expansion of the blue sensitive opsins, and also the expansion of the ultraviolet sensitive opsins, having 4 copies of the UV opsin that makes them the largest expansion so far described in insects. So looking at the evolutionary scenario of these expansions and duplications, so what we saw is that the long wave sensitive opsin is a duplication that is ancestral to all mayflies, but by contrast, the ultraviolet duplications are ancestral to the Bettidae family. And as if you remember from my first slides, so the most striking trait of the Bettidae family is the turbanate eye. So by then we hypothesized that probably there was a relation between these duplications of new opsins with the origin of this new visual system. So an easy test to have a first approximation of this was to check the expression of these particular opsins between males and females. And we did differential gene expression between the heads of the adults. And what we saw is that the two genes that are most upregulated in males are one of the blue opsins and one of the UV opsins, showing that these opsins are male specific, but are they turbanate eye specific? So they are. We check performing in situ hybridization, and so we saw that the UV opsin 4 is only expressed in the turbanate eye of the males, but is not expressed in the compound eye of the males or the females that we used as a control. So what is the function of this? So why these specific opsins and why in the turbanate eye? So we don't know yet. So this is something that we are currently working on, and is so that probably, so our hypothesis now is that probably so these opsins are used by the males to detect the females in the swarm, because mayflies mate while flying in swarms, so the males have to be able to detect the females. So what we did was to put some of our females under the UV light. And what we saw is what you can see here is that under the UV light, the eyes and parts of the cutica really bright up. So this is quite a suggesting result, preliminary result, I have to say. And especially because when we do the same with sexually immature sub-imagos, we are not able to see anything. So probably these opsins are used to detect the females that are really ready to mate and not waste the energies of the male trying to mate something that is not going to be ready for that. And finally, I just want to, as I said at the beginning, mayflies are key to understand the origin of wings in insects. And for that, we perform a different type of analysis, the clustering analysis, WGCNA. And which WGCNA give us is clusters of genes that are expressed in a tissue-specific manner. So we did that for the mayfly and also for Drosophila. And then what we made was pairwise comparisons between the modules of the two species to see how similar or different they were. And here you can see the results. And something quite nice and interesting was that the genes that were associated into the wing cluster in mayflies were really similar or related to the genes that were located in the wing disc of Drosophila melanogaster, suggesting or indicating a conservation of the transcriptomes of these two species. Moreover, so we have 126 ortholog genes that were shared between the two modules, and some of them have already a known function in wing development, but there were others that not. So we tried to investigate a bit more what happened with these ones with a known function, and for that we did a knockdown experiments in Drosophila using the UIS GAL4 system, and when we knocked down those genes in Drosophila, in all of the cases that we checked, we observed wing phenotypes, suggesting that not only is a conservation in the transcription of the genes, but also there is a conserved function of those genes. So it's a great tool to infer function of new genes. But of course, so this is just about conservation, but how wings originated the first time. So there were several, or there are several hypotheses still in discussion, and I don't want to go into the details because of the time. But so one of the hypotheses suggests that in mayflies, wings and gill plates are serially homologous organs. So we wanted to do a preliminary and first transcriptomic approximation, and for that we did a cluster of all our transcriptomes to see the relationships between them, and we observed that indeed wings cluster together with gills. So this is good. So we performed an additional experiment, which was to check genes that were highly expressed in wings and in an additional tissue. And for the 98 genes that we found that were highly expressed in the wings, 42 of them were also highly expressed in gills in comparison to the low number of other tissues. So this preliminary data really suggests that they share or they have common genetic programs, and that probably, so we don't know yet, we have to explore a bit more into the details of this, but that probably they really share a common origin. And with that, I want to finish thanking Fernando Casares and all the lab, especially the people, the students that are now working with different aspects of mayflies. And also, as you can imagine, so this is a big genome project. So even the genome is quite small. But so there were many people involved in the project. So I want to thank them all, the funding bodies, and for your attention. Thank you.
Thank you, Isabel. Really enlightening talk. I enjoyed it a lot. So we have tons of questions and only time for a few of them. So let's start by the first one. Jay Alves is asking, is it possible that the odorant binding proteins are performing something unrelated to smelling when expressing in the gills?
Well, we don't know because, of course, so this is just transcriptome data, so we don't know anything, especially about the function. But it's true that it has been suggested that odorant binding proteins have different functions beyond the perception of odors, and it's also true that in the case of Cleon, so they have many, so they have 191 different odorant binding proteins, and they don't have the same number of odorant receptors, which in theory are the ligand receptor to bind. So probably they are doing different things, and actually we are not seeing so many odorant receptors expressed in the gill. So, but yeah, I cannot say anything about the function.
Interesting. Next question by Jocelyn Gutierrez Guerrero. Did you find pseudogenization events in the opsins and olfactory receptor genes?
Yes. Well, for the odorant binding proteins, so we have 191 plus some that are incomplete. So we are not sure if they are pseudogenized or that the annotation, so the sequencing is not good enough. Even I think it's good, but so it could be. For the opsins, we found that there is one of the blue opsins that is a bit weird. So it's not a pseudogene because it is high, so the expression is really, really high. But something, so when we translate this opsin, that is not a canonical opsin. It's a bit weird. Mm-hmm.
And perhaps the last question is also related to opsins by Irepan Salvador Martinez, who is asking, have you estimated selection on genes, for example, the opsins specific to males?
No, we didn't have the chance to do that. But yeah, so it would be good to do something like this. Mm-hmm. So.
All right, so I think we should move on with the next speaker. Thank you, Isabel. It was great. And I will, so just